see two people. Number one is a famous marathon runner. His name is Jim Fix, who was running marathon at the age of 42. He started running marathon at the age of 42. The reason being his father had a genetic predisposition that he died at the age of 45. So he wanted to beat that genetics part. So he started running a marathon, so which is supposed to be a healthy endeavor. But to the astonishment, he died at the age of 43 while he was running a marathon. So this is story one. There is another person who is there, her name is Jean Calmet, who is a documented centurion who had lived for 100 years. And when you just see the lifestyle of that lady, she lived for 102 years. She's a chronic smoker since 40 years. So I'll just put a question to you here. What is the problem here? Or what is the good thing here? A person who's running marathon wanted to beat the genetics, did not beat the genetics. A person who is genetically made up in such a way that even chronic smoking did not kill her. So people started delving into a sequence of events, and people started working on the animal model and to see which are the animals that were living quite longer in their lifespan. So people moved on from genetics to epigenetics or the micro environment where they wanted to see, and ultimately they zeroed on microbes. And you all know that both good and bad microbes do exist, and they are in and on the system. So this is a recent study about the human microbiome where people had done an extensive study on the immigrants in the United States, where you would see that people, when they are there earlier, when they moved to the island, the continent there, America, there are no Native Americans. Now we call it as Red Indians. You know that Columbus, when he went there, he saw people and he thought that he went to India, but then he called them as Red Indians. So the microbiome of the pre-immigrants versus the European immigrants, who are currently there, have been extensively studied, and they saw that the diversity, the gut microbiome diversity and the overall microbiome diversity was slowly on the narrow pace. So from the diverse part, they were going to a narrow thing. And there were a lot of studies that were being built on studying different microbiomes. One among them is gut microbiome. So now we come to a controversial aspect of microbiome versus antibiotics, where when we take antibiotics to kill the bad bacteria, many of the dead bacteria do go, and because of our scrupulous use of antibiotics, still more problems do exist. And it is estimated that to 2050, the maximum killer in the whole world will be antimicrobial resistant. And this is being very much proved here since 1928, when Alexander Fleming first explored the penicillin, until the recent innovation of this NDM, New Delhi beta lactamase, highly resistant bacteria. Everything is only converging towards chasing the wind of the antimicrobial resistance. So these are all a couple of universities, academic universities, which are totally delving into an extensive study of microbiome. And with due respect to the Brits here, we have the Imperial College of London there. So to say that a lot of people have been just jumping into the bandwagon. So now, what is the real global scenario? People started studying tribal people, diversity of microbes here, gut microbes, and I'm going to tell a story at the end where our collaborator from Harvard did a very extensive study on the tribals to identify what is the best microbiome in the whole world, so that we could comment. Right from studying gorillas to chimpanzees to the humans in South Africa, they found that the diversity is a very big problem. When the diversity of the microbiome is narrow, then the disease aspect comes into play. So when you just see this particular graph, you would see that microbiome studies have just been moving on, climbing very high ladders. Okay, there are certain important labs in India, such as Professor Selvan's lab and other people, where they are only focused on that good high impact papers, where you would all could end up. A lot of other labs are coming up too, and microbiome initiatives have come. And that is the total microbiome map that has been currently published on this part. So 2007, when they started, you all know that this microbiome initiative started from Human Genome Project. And when people started sequencing genome, people got tired and they wanted to move to microbiome. And everybody jumped into microbiome, everybody did microbiome work. Then finally they found it was very, very costly because you need a gene sequencer and other things. People forgot that. And now since sequencing is cheaper, now people are again jumping into that part. Europeans in 2008. Ultimately, what you would see here is the antimicrobial resistance or obesity or type 2 diabetes. Everywhere they could find only they would relate to microbiome. So this is not a story that I'm cooking up. There are evidences to show you that. A couple of things which I will also show where microbes predisposed to many hereditary diseases as well. So this biome is a flooded species of energy where they culminate into many small molecules. So these small molecules are the focus of my talk here, where what we could do as an alternate for antibiotics with the microbiome person. So taking this life expectancy map here, you would see there are certain European countries where it is more than 80, and African countries which are less than 40. But ultimately, when you see this map, there are 5 places in the whole world where people live more than 100 years, centuries. And one of the places, the place where I just come from, this is Loma Linda, California, this place where I hail from, where I was working and still working for 18 years. So what is so beauty about that? People, they live average more than 100 years, centuries. So now the National Institutes of Health had poured on a lot of money to study what is really, really good to that. And a part of that was actually allocated for the microbiome study. And I should say that this is not a rat study, okay? This is a human study, okay? This is actually following the human being for a period of, say, 30 to 35 years. And you could see here, I think the graph is not that much visible, but you would see here from which year it started. You could Google that, you will see evidences of that part. So people have focused on to certain important bacteria which live in our system, which are very important for longevity and for the gut health and other stuff. So going to that good bacteria, which we call it as probiotics, there are a lot of alternatives to antibiotics which have been focused on. But ultimately they land up only in microbiome as medicine. Right from small molecules which are now catching up very, very, very good in the system. People are just focusing on many, many initiatives to satiate the hunger of alternating antibiotics. I don't want to go very deep into that. Antibodies are being used, and there are a lot of other alternatives that are being used, but nothing is proving well. And people have just moved on to lysines. And they did a cocktail of lysines, which we call it as the anti-lysines here, where you would see here, this is a lysine part, where when a bacteria is being put to the effect of a lysine, you would see that it takes, say, quite a long period for the bacteria to die. But on the other side, if it is going to be an anti-lysine, which is a cocktail of enzymes that are being taken from the bacteria, probably good bacteria which are in the microbiome, and then you add up with another cocktail. What you would see here is, you could see here that the anti-lysines could kill these bacteria very, very fast. So this was also catching up in the research system, but finally people have focused on ending up in antimicrobial peptides. So what are these antimicrobial peptides? So as you know that, as I've already told you, biome is flooded with energy, and each microbiotic species, they end up in transformation leading to new molecules. So when the bacteria in your system is going to interact with the host, they are going to bring about new molecules. So this is where my focus of the study is. We are focusing on these new molecules with such a lot of interactions with the microbiome, with a slew of bacteria or slew of microbes inside, where you would see here when you have 10 trillion human cells, you have only compared to 100 trillion microbial cells. It means to say that the red part is only the human being, and the white part is only the bacterial or the microbial part. Taking away a comparison with the genes here, 20,000 to 30,000 genes and still counting, versus the number of genes of microbial origin are 2 into 20 million. So now see the gene interaction part, protein interaction part. So ultimately what really happens is, this is a diversified different kinds of microbes, fecal to oral to skin and everything. And they found two important microbiomes in the human. One is oral microbiome, which is in the mouth, and the other one is the gut microbiome. So microbiome helps in the prophylaxis concept where you would see here. I don't want to go and do a theoretical point of it, but I will say that when there are certain good bacteria, they take care of the bad bacteria. But at the same time, there are important bacteria which are called as keystone pathogens, which are very, very important, play a pivotal role in the total dynamics of the microbiome. They move from symbiosis or a common healthy state to a dysbiotic state where the host itself is predisposed to many diseases. Why is just because that each and every bacteria that are in the system produce one way or other important unique molecules. And these molecules interact and with the host and ultimately do a cause effect with the host. Okay, I'm just showing you here top tier publications which have come since 10 years ago. This is on the oral microbiome, where people are telling that bacteria in stroke and heart disease. They have identified bacteria which is in the mouth. So it means to say that if I will tell you, if I'm going to tell you that if you don't brush your teeth well, you will die of heart attack. You will laugh at me, right? But this is what these publications are telling you. Yes. Okay, as I was just telling you today morning, if you eat chocolate, you will get Nobel Prize. But now, these are all top tier journals. They are telling this. Okay, then what is this? People are also comparing human microbiome to many diseases like diabetes mellitus, Alzheimer's. See, American Society for Microbiology 4 years ago have highlighted a bacteria in the mouth and moving on to the stomach called Papillomatosis gingivalis that causes Alzheimer's disease or predisposes to Alzheimer's. So this is one of our publications where we did a very extensive study taking samples from different continents. You would see here we have just compared with the genome database from two different databases. We found a lot of confusions here, okay, especially with the Asians, where we found a total change in the picture only compared to the Asians. As I've already told you, our microbiome got very confused itself as compared to the other things. But microbiome study itself is very confusing. But we wanted to just bring in and focus on what unique bacteria cause a shift from healthy to disease. So what you see here is the blue one. This is a panel which is being a group of bacteria which is being, I don't think it is very clearly visible, but what I'm telling is this is the phylum Bacteria and Bacteroides and Fusobacteria. And these two are very much highly upregulated in disease conditions. And we wanted to focus a representative sample. We narrowed down to two different bacteria, which are Papillomatosis gingivalis. And this Filifactor allosis is a very unique bacteria, which our laboratory, where the initial people who we delved into this research to identify them as a keystone pathogen. So these two are published in the cover page of Infection and Immunity. And what you see here is a human cell that is being eaten up by these two bacteria on the right side. And what you see on the left side is Papillomatosis gingivalis. Okay, so this is an electron micrograph. Imagine with all these 20 million bacteria inside your system. Imagine I have taken only two bacteria, which are keystone pathogens, keystone or king pathogens. When you put them in vitro, you will see here this is a single cell and the V stands for vacuoles. These are the vacuoles where both the bacteria are very much covered. So to say that if one is there, it is very quiet and normal obedient bacteria. When two bacteria are there, then comes the problem. It is like our own students, right? So when one is there, people are quiet. If two people, then we become so mischievous. Like that the bacteria do. But the problem here is these two are keystone pathogens. So we did a quantitative phase contrast microscopy of single cell to a single bacteria. What you see here is an unique time sequence, time course sequence. This is the start here, this is the end here. Ultimately, the cell gets totally killed, disintegrated by these two bacteria. So we also proved them with a time course delay, heavy high tension transmission electron microscope. You see this time course starting from A, and this is the end of it. You see this eukaryotic cell, human cell. It is not there here. And this is only two bacteria. Imagine in our system how many bacteria are there. So this is an insect. If one bacteria is there, this is the human cell, there is no change. If two bacteria is there, this is Papromonas gingivalis, and this one is, this is Phylifactylosis, and this is Papromonas gingivalis. One is gram negative, another one is gram positive. So we did a global proteome analysis. We did a very extensive proteome metabolome study to study the path. To make the long story short, all the atrocities happen in the cell death, cancer pathway, okay, upregulation of cell proliferation, cell death or apoptic pathways have been highly upregulated. But we do not know, but we harbor this bacteria in our system. So we did a network analysis. This is a bioinformatic analysis. We identify what are all the proteins that are highly upregulated in rheumatoid arthritis. The reason is, one of our PhD students, you all know that students are very clever than the teachers, like me. So she came and told, why don't we do a bioinformatic analysis to see what are all the proteins that are highly upregulated in cardiovascular disease, rheumatoid arthritis, diabetes mellitus. Okay, go ahead. So then she came out and she did a very extensive study where she identified rheumatoid arthritis protein, which have been highly upregulated, and these are all published data. But whereas this is the data, proteome data, which is clearly corroborating with the result. So the story here is simple, that if you are going to put these two keystone pathogens together, the host predisposes to rheumatoid arthritis. This is one of the earliest seminal publications of microbiome. This came in 2006. People started telling that when there are certain groups of bacteria in your system, you become so obese. Okay. And there are certain people who will eat tons of idlis, but they will be very lean. You might have seen within your own friends. But there are a couple of people, when they eat 2 idlis more, they will become so bloated. So why is just because, this is our author here. His name is Lannan. So he is the one who published this in Science, where you would see here he is very, very fat in nature, and then he is very thin here. So, yeah, mice experiment versus a human experiment. So they found that there are certain unique bacteria, and this is one of the recent concepts that is currently up and running with the Japanese group. Now people have isolated this Christentia minuta, which is very much highly there in the Japanese population, which is the reason for people who are thin and Japanese person to be very bloated, very, very lean these people will be, right? So if there are any Japanese or Chinese here, my sincere apologies, but I'm just telling you, it's a good advantage. So since then the sequence was there, we also wanted to do a study. So we delved into the Indian population here, and the study is currently underway with CCMB Hyderabad, where we have collected the samples, and we wanted to see what are all the unique bacteria that are going on. And this is going on a very snail pace, but it's still going on. We identified differentially expressed conserved microbial communities. The bacteria communities which you see on the red are the communities highly upregulated in disease condition, no matter what the disease. And the blue ones are the healthy bacteria. There are in between that black bacteria, that is the culprit here. They are very quiet, but based on the shift, they can be either blue or be red. So we did a metabolome study here, and I do not know you will be able to see this Odoribacter. Odoribacter is a very unique bacteria we found in 4 different ethnic groups in the whole world. They can produce butyrate in a high way. Okay, what is this butyrate do? This is a cycle metabolic pathway where I am just elaborating to tell that the high production of butyrate is here. And this is being facilitated by this bacteria. And it is just because it is totally modulated by the diet here. Okay, coming to the diet here, 75% of the plant genetic diversity we totally lost in the whole world. Now, if you imagine if you are a non-vegetarian, you only take either chicken or mutton, right? So maximum 6 livestock breeds is what we eat for a non-vegetarian meal. And if you are a vegetarian, you are only eating 12 species or a group of plants. Versus 250,000 and odd plant species. So that is why, we are coming to another story at the end. I'm just telling you, I'll corroborate this one. Imagine with this, you don't get a diversity in your gut or in your mouth. So we very extensively studied what are all the things that you see here are different unique microbiomes, right? From skin to saliva to dental caries to gut microbiome to the fecal microbiome, everything. And what we studied was certain interactions have brought in some small molecule protections, which are very good alternatives for antibiotics, like antibiotics, bacteriocins, microcins, etc. And these are a couple of bacteria which I am just showing you, and the small molecules which are antibiosis in nature. So we did a genomic approach versus a metabolomic approach, approximately with all these 250 compounds that have been identified. We wanted to zero it down to identify some unique small molecules which we can take them and use them as an alternate to antibiotics. So what we did was, we took the microbiome from the human being and we did a culture of that. And the culture is super native. We identified whether we have small molecules or not. Okay. So you might ask me, how can we narrow down from such a big, vast crowd? So we reduced them based on the prevalence rate, based on the previous publications. Right? So we came and we did it. This is a mass spec analysis here. So we grew them on a in vitro analysis. Supernatant we have taken. We have did a mass spec analysis, and then we also did a LCMS with a very extensive bioinformatic analysis. We identified that there are certain interactions which bring about unique molecules. You don't see them. I have not given the legend here. These are all interactions with the Streptococcus mutants and Papilloma gingivalis, which are in violet in color. There are many bacteria which I am going to tell you now. These are all unique stretches of amino acids that you call them as small molecules. We have identified them based on the spectrum. So we did a bioinformatic analysis to identify the spectra that we got are small molecules. They belong to these different types of alternate to antibiotics. So the microbiomes, we took them, we interacted them with the host, we got the small molecules, we identified the small molecules to be some unique way. And what are they? These are all secreted small molecules, and what you see here is a bacteria, and what you see here is a small molecules which you have identified by the previous experiment which we have already told. So the take-home message is so simple. If you have the bacteria, and if you have the bacterial interaction, the bacterial interactions can be detrimental as you see here. They can kill your cell. But at the same time, if you are going to just grow them in vitro and identify some unique molecules which are being produced by the bacteria to defend itself, and if you are going to explore that as an alternate to antibiotic, you could use these microbes as a probiotic nature. And you can use them as a probiotic instead of using the whole bacteria as we use in probiotics, like lactobacillus. Rather than, we can isolate the small molecules and we can use them as an alternate to antibiotics. So, coming to the diversified the microbiome. So this is a Harvard-based study with a lot of collaborators here. So this is the Hazda tribe of Tanzania. So people took tribal population because they are the native virgin population that you would ever see. Okay. For example, imagine if it is going to be India, we go to Andaman Nicobar Island where there are a lot of tribals there. We take samples from them and we get unique things like that. We also did it in Hazda tribe hunters. These people are called Hazda tribe hunters. And what you see, these beautiful people here. They are indeed beautiful just because their microbiome are very diversified. Okay, they eat whatever they want to eat, as you see down. Okay, they take honey, they take herbs, they take everything. They don't take bhaji, bonda, vada, and ultimately they have a very good microbiome. Okay. So, having said that, the small crux of the message is so simple. Microbiome is very diversified, and we have a lot of bacteria and viruses which are there. Not even many are being classified. But at the same time, there are a lot of pioneering people who are just coming up with microbiome research. Okay, like British people, UK. So we have a lot of stalwarts there. Ma'am is there. She's going to tell you about her infectious host pathogen interaction part, right? So you should totally understand that we learn from the British earlier, right? But I should say one funny story that once Sir Isaac Newton, who's a British physicist and a mathematician, who traveled on the flight with one of the Indians, and since their flight is 14 hours, he ended up with a conversation, and they wanted to have a puzzle resolved. So one will ask the question, and if the other person does not tell the answer, he will give 5 rupees, and vice versa. If it is going to be Isaac Newton, he'll give $500. Okay. So then the first question was shot by Sir Isaac Newton. What is the distance between Moon and the Earth? Our Indian did not know anything. Probably an Indian student, though. Probably. He might not know anything, so he said he gave 5 rupees. And the turn is now Indian student, and the Indian student started asking a question. Which animal goes up the hill in 3 legs, comes down with 4 legs? So, with all possible knowledge, Sir Isaac Newton got very confused, being a great mathematician, physicist, everything. And finally, he gave $500. So, before going to the next thing, Sir Isaac Newton asked him, okay, fine, I gave the money now. You tell me the answer. Our Indian gave 5 rupees back and told that I do not know the answer. So, what I am coming to say is, even though we are being ruled by the UK, and even though finite by us, it is our Indian minds who are there everywhere doing such a lot of things. And microbiome research is an upcoming field where I'm indeed very happy that Professor Selvan's lab is being delving into this very sincerely. Thank you very much.